Hi, welcome everyone. So Tuesday, 5 o'clock, once again, another exam paper to go through. And today we are doing the AQA 2017 paper, paper 2. And I am just now suddenly doing a quick check to make sure that everything is set up, and let's hope that that is the case. So without further ado, I'm going to go through questions. I'm going to show you the kinds of answers that I would have given. And then I'm going to talk through the mark scheme and the marks that I got and the marks that I failed to get because I didn't get them all. And probably worth just pointing out to those of you who are aiming for those really high grades, don't panic if you don't get every single mark on the paper. Yes, of course, you need to be sure that you're going to get enough marks to get the grade that you're aiming for. But you can drop probably at least 20% of the marks on most papers before you are in danger of losing your A star. So don't get hung up about every last mark. You will kind of notice that I'm not getting hung up about it when I lose marks because I know that I've got enough in the bag, as it were. Okay, anything else that I need to say? I don't think so. Let us just get on with this. So, question 1, exercise causes an increase in heart rate. Describe the role of receptors and of the nervous system in this process. So here we are talking about exercise. Of course, if your muscles are exercising, they're going to be producing more carbon dioxide. And as a result of that, the pH of your blood is going to go down because it's going to become more acidic because the carbon dioxide reacts with the water to make carbonic acid. That's why the pH goes down. Remember that those pH numbers at the lower end of the table is what, at the lower end of the scale is where the acid is. Alkaline, of course, is above 7. And the decrease in pH is detected by chemoreceptors. This causes the medulla oblongata, or just the medulla, to increase, now here's the important thing, to increase the frequency of impulses down that sympathetic nervous system, that sympathetic neuron, to the SAN of the heart, to increase the heart rate. So, of course, that the blood can flow faster to the lungs so that you can excrete that carbon dioxide as you're breathing out. Hi, Lavinia and Samantha. Hey, Samantha, we're going to go through the mark scheme. So you're going to see what I have written. So I have not just rewritten the mark schemes. I have, when I prepare this presentation, I'm literally copying and pasting all the questions, and then I just copy and paste all the comments. I'm not sitting there reading them. And I consciously don't read the mark scheme while I am writing my answer, because I want you to see what I would actually have written. So there is going to be a difference, but hopefully I'll explain, Samantha, why certain things get marks and why they don't. Terminology is key. Anyway, let's keep going. So you'll see, because I remember now, on this one, I didn't get all 4 marks. Here is the mark scheme on the left-hand side of the screen. And here are the notes of the examiners on the right-hand side, what is allowed and what isn't. And this is where I got the marks. And notice that I number my marks according to the numbers of the mark points on the mark scheme. So you can see exactly where I achieved the mark. So I got mark points 1 and 4, and I think I got another one. Yes, I got mark points 1, 3, and 4. Now, I could kind of kick myself for not getting mark point 2, and I'll tell you why. Because the question was clearly about, the emphasis was on the roles of the receptors and of the nervous system. So the fact that I didn't talk about the neuronal connection between the receptor and the central nervous system, the medulla in the base of our brains, I was kind of going, oh, duh, you know, that was a bit daft. That was a silly mark to have lost. And it was because I didn't read the question carefully enough and take on board that the emphasis was on receptors and the neuronal communication. So my fault. Okay, let us move on. AMP, activated protein kinase, oh, thanks for that name, is an enzyme that regulates a number of cellular processes. Exercise leads to an activation of AMPK. And this figure shows you what AMPK does. So AMPK inhibits the production of malonyl coenzyme A. Okay, so here I am. Exercise is the notes that I would have written on the exam paper. Exercise increases AMPK, which stops this pathway here. Now, that of course means that you're going to have a decrease in malonyl coenzyme A, which is therefore going to decrease the inhibition of this pathway here. So if you've got less of malonyl coenzyme A, you're going to have increased conversion of fat or transport of fatty acids into the mitochondria. Hopefully that's me just deciphering what the question is kind of telling us. And this is me saying increase fatty acids into the mitochondria. So CPT1 is a channel protein that transports fatty acids into the mitochondria. Fine. Using figure 1, explain the benefit of activation of AMPK during exercise. Well, my little notes here have kind of already answered the question. So AMPK causes a decrease in malonyl coenzyme A. This would therefore increase the transport of fatty acids into the mitochondria. Now, here's the key. The activation, just reminding myself, this is the beginning of the question and exercise is the end of the question. So I'm starting off by saying the activation of AMPK causes a decrease. That is the start of the story of my answer. But the end of the story is when I get to relating it to exercise. And here is my absolute tip to you. Whenever you see the term exercise in a question, think muscle contraction, ATP, respiration, because those are often mark points. So notice the pattern of my answer. Increase the transport of fatty acids into the mitochondria where they can be used in respiration to produce ATP for muscle contraction, which is exercise. Hopefully that makes sense. And as a result, I think I got the 3 marks. So 1, 2, respiration, yes, can be used, yes, for muscle contraction to produce ATP. It would have been really easy to have lost this 3rd mark point, the 3rd mark, if I hadn't made that kind of mental connection that exercise equals muscle contraction, which requires ATP from respiration. All right, so here are all of the alternative things that you could have written, but hopefully that makes sense. Oh, suddenly just realized we've got some. Oh, thank you, Daniel. Samantha, yes, this video always goes onto the channel. So yes, and the joy, if you are watching this in non-live stream land, the great advantage of doing that is that you're going to be able to stop the video, you're going to be able to look at the question yourself, see what you would have written, and then you'll see what I would have written and what I would have got. So there is almost an added benefit, but this is why I'm going through it really quite quickly. So those of you who are watching live, I'm trying to do this as efficiently as possible. Dengue, serious disease caused by a virus. It's carried by the mosquito Aedes aegypti, I think. One method to try and reduce transmission is sterile insect technique. This involves making the male mosquitoes infertile using radiation, basically irradiating them. Explain how using the SIT could reduce transmission of dengue. So this is what I wrote. Sterile male mosquitoes will be unable to reproduce, therefore reducing the number of mosquitoes over generations, gradually dropping the numbers. Here's the mark scheme. And of course, I only got mark 0.2. I completely forgot about the whole competition thing with obviously non-infertile male mosquitoes. Hey ho, that was a bit silly. Again, a bit of a silly mistake. I'm going to be honest though. So here, when I look at that, I just know I wouldn't have thought. It wouldn't have been at the top of my brain. I wouldn't have written it. So it's one of those marks that even though I knew it, and there's a bit of me kicking myself about it, I'm also kind of going, do you know, I just wouldn't have thought of it. I think I'm okay with the marks that I've got. So, and let me endorse that attitude. Describe how the Mark-Release-Recapture method could be used to determine the population of the mosquitoes at the start of the investigation. Here we are, just going through the Mark-Release-Recapture. Notice that we have to describe the method to the point of determining the population size. They want to know how you're going to get the number. Hence, this bit at the end, the calculation, okay? So, sample of mosquitoes caught, marked, and released. Here am I know what's on the mark theme for this. The marking must not be toxic or increase predation. Yeah, it can't cause them to die. It can't cause them, can't cause any reason to die. Suitable time is left before capturing sample 2 so that the released mosquitoes can randomly mix in the population. Then you have to count the number in sample 2 and count the number that are marked in sample 2. And then you use this calculation. Population estimate equals the total of sample 1 times the total of sample 2 divided by the number marked in sample 2. Here is the mark scheme. And here we go. And there's the mark. So I got all 3 marks. But notice how actually getting the number is the end of the question, and therefore that was my prompt. I kind of got to the end here and thought, I've got to get my calculation in. But I didn't know, can I just point out, these are sometimes when you're asked to do a full-on description of Mark-Release-Recapture, these things that I have written here, this, like the marking mustn't be toxic, absolutely can be a mark point in itself. So hopefully that is helpful. The release of radiation sterilized mosquitoes has not been very successful. One reason why? Well, radiation isn't generally very good for any living organism. So this is what I put. Notice that it was important that I put that it kind of leads to death before any mate. No, actually it doesn't matter, does it? Because they're flipping infertile. I'm being a dipstick, ignore me. Okay. Oh, well, I didn't write that. Oh, it affects their lifespan. So I got the mark because of lifespan. Yeah. Okay. So suggest why the scientists released more. Oh, sorry, I should have said here. New method. They produced transgenic males. Transgenic means they put a gene inside them from something else, carrying a lethal gene that kills the offspring before they can reproduce. So these males can do all the reproduction they like, just the kids are going to die. Sorry, that's not very nice, is it? Scientists, they release them every week in the area of a city in Brazil. regular intervals, they determined the number, and they compared with a control area where the transgenic mosquitoes weren't being released. I must admit, my first thought was, mosquitoes can fly, so they must have been somewhere quite distant, I'm assuming, but I'm kind of bearing that in mind. Suggests why the scientists released more transgenic males each week. Well, that's because every day some of these mosquitoes are going to die. And maybe that's the bit of information that actually isn't strictly in the A-level biology syllabus. It's not like you have to know that mosquitoes have a very short lifespan, but they do. They don't live for long. So I must admit that to me, it wasn't too hard, but I would understand for a 17, 18-year-old that might not be obvious. I get that. Okay, so I got the mark point there. The release of transgenic males proved successful in reducing the number. Describe how the results support this conclusion. We are basically looking for evidence in the graph. What is it about the graph that says, yes, this has been successful? And to me, clearly, this downturn here really is very obvious. It kind of goes down, it goes up a bit, and then it really comes down, and importantly, stays down. And this is in comparison to the control area, which you can see fluctuates a great deal, but there is really a significant difference at this end of the graph. The numbers have dropped and they stay down. So this is what I put. In comparison to the non-treated area, by 12, 13 weeks, I wasn't quite sure what to put, 12, 13 weeks, there was a significant and sustained decrease in mosquito numbers. And I thought, when I looked at it, I thought, crumbs, there's less than 100 per kilometre squared. That is not very many. Here's the mark scheme. And here are the examiner's comments. So I actually ended up getting mark points 1 and 2 with what I said. And actually, bizarrely, the number of mosquitoes is lower after this number of weeks. So, and if I'd only talked about there'd been a decrease without the reference to the number of weeks, I would have just got the 1 mark. So, okay, question number 3. Scientists investigate the effect of regular exercise on skeletal muscle fibers in mice. Scientists compared the muscle fibers of mice after 6 weeks of regular exercise, trained mice, with those of mice who had not exercised, control mice. The scientists stained the muscle fibers from both sets of mice to show succinic acid dehydrogenase activity, at which point your brain's kind of going, whoa, never heard of this. The darker the stain, the greater the succinic acid dehydrogenase activity. So we're kind of going, okay, trained mice, more of this succinic acid dehydrogenase. And then we're told this is an enzyme used in the Krebs cycle. And now, as soon as I see the Krebs cycle, I'm kind of going, that's aerobic respiration. Yeah. So I'm looking here and I'm kind of going, okay, these muscle fibers are doing a significant amount more aerobic respiration than these control mice. And my brain is now going down the route, slow twitch muscle fibers, slow to tire, use oxygen, lots of very much redder because they are infused with blood vessels, etc., etc. So suggest one reason, and myoglobin, all of that kind of stuff. Suggest one reason for the difference in the staining between the muscle fibers of the control mice and the trained mice. So I kind of went down the route of, oh no, I didn't. I thought I did the whole slow twitch thing. more aerobic respiration is occurring in the trained mice muscle. So, and there's your mark point there. So I've got the mark. The scientists then compared the length of time that the control mice and the trained mice would carry out prolonged exercise. The trained mice were able to exercise for a longer period of time than controlled mice. Explain why. Okay, here's my little comment on explain why questions, particularly if they are 3 or 4 marks. So we are now wanting to use our A-level biology knowledge to get me the marks. So I'm immediately kind of thinking, okay, this is because that they can do, my brain went down the lines of, okay, they can do more, they can do aerobic respiration, they can do more aerobic respiration. That, of course, here we are, exercise, that, of course, produces more ATP, and therefore the muscle fibers are able to work for longer. And that is, I think, pretty much what I put, which releases more ATP for muscle contraction. Notice that my brain is doing that whole link thing between exercise, ATP, and muscle contraction and respiration. Than anaerobic respiration in fast twitch. This means they can exercise for longer. So this is the mark scheme. So here was me. Now, I did feel a little bit silly on this. I only got 1 mark out of 3 because I was totally going down the whole aerobic respiration is the more efficient form of respiration, produces more ATP. Hey, you know, easy peasy lemon squeezy. I completely forgot the other half of the explanation, as it were, because in what is happening in those control mice that haven't been exercised, of course, they are not able to do all that aerobic respiration, and therefore they're going to be doing more anaerobic respiration, and therefore producing more lactic acid, which is going to really tire the muscles more. I completely just went down the route of aerobic respiration rather than what is also happening in the control mice group, which was a bit daft, to be quite frank. Okay, but moving on. Scientists determined the mean diameter of muscle fibers in trained mice using an optical microscope examining sections of muscle tissue. The circular area of one field of view was 1.25 millimeters squared. The diameter of this area was equal to the diameter of 15 muscle fibers. So here am I just imagining 15 muscle fibers across the diameter of a field of view. Field of view being that circular picture that you see down a microscope. It's not a square, it's a circle. Hence the use of pi r squared. Using this information, calculate the mean diameter in micrometers of muscle fibers in this section of tissue. So this is involving rearranging of equations. Pi r squared equals 1.25 millimeters squared. Therefore, r squared equals 1.25 divided by pi, which equals this. When I do the dot, dot, dots, it means I can't be bothered to write out everything else that is on the calculator. I have not rounded this last number because I am saying this should be held in your calculator. Don't just, you know, round it up. Then, so r is this, but I'm wanting the diameter. So of course I've got to times the radius by 2, which equals this number going on in the calculator field of view. Okay, and then I need to divide that number by 15 because of course that diameter is the equivalent of 15 muscle fibers. And therefore I get 0.084 millimeters. I've now got to remember to convert this into micrometers. Of course, there are 1,000 micrometers in 1 millimetre. So if I've got 0.084 millimetres, I'm going to have 84 micrometers, moving the decimal place 3 places. Okay, there's your mark scheme. Here are the notes, and I get the 2 marks. Please do chuck in any questions if you have them. Very happy to deal with them. There is a bit of a delay between you seeing me and me speaking, so just so that you're aware. So the scientists also compared the diameter of samples of muscle fibers taken from young mice and adult mice. Here's the graph. Describe 2 differences. Well, the obvious thing to me, number one, the range here of the young mice is much less than the range of the older mice, and actually the peak is higher in the, where the peak of the modal, it is the modal number, is higher in young mice than it is in old mice. I wasn't absolutely sure that I didn't need to add numbers, so I did. It was one of I literally wrote the 2 sentences first. I wrote the range is smaller than young mice than in old mice, and the mode is higher in young mice. But I then kind of thought, that feels too simple, so I put the numbers in. Here's the mark scheme, and I found this a little bit bizarre because I put these numbers in, I actually got marks 1 and 2 here, which I found a bit funny. I think if I hadn't put any numbers in, comparison of the range without numbers, I would have got mark 0.2, and I would have got both marks actually if I hadn't put the numbers in, but it just felt a little bit simple to me. Question number 4, isolating chloroplasts from spinach leaves to form a chloroplast suspension. He used the chloroplast suspension and the DC-PIP solution, here we go, required practical, to investigate the light-dependent reaction of photosynthesis. DC-PIP is blue when it's oxidized, but when it gains electrons and is reduced, reduction is gain, it goes colorless. So student set up 3 test tubes. He set up a tube with 1 centimeters cubed of solution without chloroplasts, so just the buffer that the chloroplasts were held in, and 9 centimeters cubed of DC-PIP solution in the light. No chloroplasts, DC-PIP. Tube 2, chloroplasts and DC-PIP, but in the darkness. So I wouldn't be expecting this one to change color. I wouldn't be expecting this one to change color because there's no dark, there's no light. And tube 3, chloroplast, DC-PIP and light. This is the one I'd expect to change color, which is what is shown in the table. The solution that the student used to produce the chloroplast suspension had the same water potential as the chloroplasts. Why is it important? 2 marks. I can almost guarantee that one of the marks will be because you have to say the word osmosis, okay? And I'm pretty sure of the second mark. So the water potentials have to be the same so that water neither enters nor leaves as a result of osmosis. Because if the water enters, the chloroplast will burst, and if the water leaves the chloroplast will shrink. And hence, that's why I've written what I have and put the word respectively on the end, so that going into is relating to burst and going out of is relating to shrivel. So here are the marks and the examiner's comments. And so I ended up getting both the marks because I said both the same things. Osmosis does not occur, water does not move out by osmosis, and it doesn't burst or shrivel. Explain why the student set up tube 1. So tube 1 was no chloroplast, DC pip, and in the light. Well, this was a control, not a control variable. A control is the part of the experiment that you do so that you can compare the results of that part of the experiment, in other words, tube 1, with tubes 2 and 3, to ensure two things. Number one, that the thing that is missing, i.e. the chloroplast in this case, are the thing that actually cause the experiment to work. In other words, it's the chloroplast that cause the DC pip to decolorize. And also to prove that it isn't anything else in that tube setup that causes the DC pip to decolorize on its own, as it were. Because obviously if that were to happen, it rather screws up the whole of your experiment. So as a control, to show that photosynthesis does not occur without chloroplasts and that DC pip does not decolorize as a result of either anything else that is in the tube, and the only other thing is buffer, or light, because that was the condition that the tube was under. So here we go, and there are the two. So this was interesting because the marks were basically for really explaining what a control does. It shows that the active ingredient of that part of the experiment is the thing that causes the measurement to change, in this case, the decolorization of DC pip. But it also shows that there isn't anything else in that tube setup that causes the decolorization on its own. I hope that makes sense. Two aspects to what a control does, worth getting your head around those, because a 2-mark question about a control will be those two things. Explain the results in tube 3. Okay, so why does the DC pip decolorize? Well, that is because the electrons from the chlorophyll are being gained by the DC pip rather than going down the electron transport chain. And therefore, the DC pip is being reduced rather than that first protein in the electron transport chain, and DC pip decolorizes when it's reduced. Okay, so, and those are there's the mark scheme and the notes, and there are marks. The student evaluated the effectiveness of different chemicals as weed killers by assessing their ability to prevent the decolorization of DC pip. Because of course, active photosynthesis decolorizes DC pip. In other words, healthy photosynthesis. Something that stops photosynthesis is obviously not going to be great for the plant, unhealthy, yeah? So therefore, weed killers. He added different concentrations of each chemical to illuminated chloroplast suspensions containing DC pip. He then determined the IC50 for each chemical. IC50 is the concentration of the chemical that inhibits the decolorization of DC pip by 50%. Explain the advantage of the student using the IC50 in the investigation. I'm going to be honest, it's kind of going, what's the great advantage of just doing it halfway, or, yeah, measuring the, yeah, working, because it says determined the IC50, it didn't calculate determined. Yeah, to be quite frank, I wasn't really sure. So I went along the lines of that it would be easier to compare the weed killers. I was not convinced I was going to get the mark for this. So easier to compare the different weed killers. I felt this was I didn't like this as an answer. I was not convinced I was going to get the mark, but I think, yeah, see, I did get the mark. I actually think that, where is it? This saves time waiting for complete decolorization. Actually kind of makes the most sense to me. But Also, understandably not very obvious. So, explain how chemicals which inhibit the decolorization of DC-PIP could slow the growth of weeds. Inhibit the decolorization of DC-PIP. We're needing to remember that this is an explain question. It's 2 marks. It isn't just going to be, it stops photosynthesis and therefore the plant doesn't grow, because that would be linking the 2 things. The key for this question is to recognize that the DC-PIP is, the electrons are being taken from the light-dependent reaction. They're not going down the electron transport chain. And therefore, to get the 2nd mark, we're going into more detail about photosynthesis. Slows the rate of photosynthesis, especially in the light-dependent reaction. If no, here we go. This is where I'm going into the detail. Excuse me, sorry. If no ATP or NADPH is produced, the light-independent reactions can't produce glucose, which leads to growth. And I'll be honest, I wanted to go on and say, can't produce glucose, and glucose is the basis of all the molecules that the plant has to make, which leads to its growth. The proteins, the carbohydrates, the lipids, DNA, everything. Here is the mark scheme. So, here was me. And it was only at the end of the 1st sentence that I decided to write the 2nd, go into the detail. So, thank goodness I flipping did. Otherwise, I wouldn't have got the marks. Arbuscular mycorrhizal fungi, AMF, are fungi which can grow on or into the roots of plants. They increase the uptake of inorganic ions such as phosphate. This is a mutualistic relationship. I scratch your back, you scratch mine. Suggest one way in which an increase in the uptake of phosphate could increase plant growth. They are wanting you to say exactly what molecule could be made with phosphate. And to me, personally, for me, the obvious one is DNA. So produce a named phosphate compound. So I got the mark. Suggest one way in which AMF may benefit from their association with plants. Well, they can grow into the roots. That means they can access the glucose or the sucrose that the plant has made by photosynthesis. So there you go. Example of a carbon-containing biological compound. In other words, you could have said anything. Except sugars, organic compounds, ignore products of photosynthesis. What they mean is, you weren't just allowed to say the products of photosynthesis. You had to name something. Oh, sorry. Oh, right, lovely. You're all having a little conversation. Okay, good. I'm pleased that this is helpful. Hey, maybe have a chat amongst yourselves on your phones, WhatsApp or something. Anyway, moving on. Oh, now I feel like a teacher. Oh, I'm telling you off. I'm sorry. Okay, I'm not a teacher. Anyway, explain why an increase in shoot biomass can be taken as a measurement for net primary productivity. I struggled with this. I knew what I wanted to say. So anyway, this is what I said. Here's a clue. If ever there is a question that talks about net primary productivity or gross primary productivity, write the flipping equation. If nothing else, that might get you a mark. All right, so just do it. So I know that net primary productivity is the growth of the plant because the glucose that has been made in photosynthesis, GPP, has been turned into all of those other biomolecules. The bigger carbohydrates, the proteins, the lipids, the DNA, the RNA, all of those things, which are the molecules that bring about the growth of the plant, which is what I kind of put. MPP equals growth of the plant, and shoot biomass is a measure of growth. GPP is glucose just produced by photosynthesis, but of course, some of that glucose is used in respiration and therefore cannot be used to make these bigger biomolecules. So I wrote this, here's the mark scheme. I completely missed the hint. Is it a hint? The word biomass, that we could have defined biomass. I know it, absolutely know it. I didn't think of saying it. So I got 1 mark. Look, here we go. There we go. Got a mark for saying that. Using the data from figure 5, evaluate the effect on plant productivity of adding AMF species and adding phosphate to the soil. So here is my full-on answer. Notice my little notes up here. Evaluate means pros and cons, fors and againsts. And it also means do the fors and againsts from what the data tells you. Look for the things in the data that say, oh yes, increased phosphate does increase the productivity, and look for things in the data that say, no, it doesn't. Then look for things in the data. And then once you've run out of things to say about the data, critique the experiment. Okay, so this is my format. So for increased phosphate, so I noticed that 2 out of the 3 AMF species increased their biomass. Look, I've told the examiner the abbreviations that I'm going to use. So I can now use SF and GC. So you can see that SF increased and GC increased. Control also increased, as you would expect. Yeah, does that make sense? If you increase the amount of phosphate in the soil, you'd kind of expect any plant to increase, even if it hasn't got these AMF around its roots. The reasons against the increased phosphate increasing productivity is because this one here, now which one was it? This one here, EI, enterophosphora infrequens, decreased in biomass. That's against this argument. And also, that really, just because I kind of look at these enough, it was patently clear to me that you could not tell whether these increases and decreases were significant because no statistical tests have been carried out. Major issue. So, and then I went on to say, now arguments for the use of AMF. So now we are just needing to look at this bit of the graph here, because of course in these plots of seed, they've planted seeds, and then they have measured the shoot biomass after 20 weeks, and they did it in 2 different series of plots, ones without normal phosphate and ones with twice the amount of phosphate. And in each plot, they sowed seeds, some with no fungi, some with, and then one with each plot of the different fungi, the 3 different fungi. Hopefully that's kind of clear. So arguments for using AMF. So here I'm just looking here. So in comparison to the control, yeah, so the control had none of these fungi with them. To see whether the fungi did anything, you would expect them to grow more than the control, just being in the ground on its own. So IE and GC increased, but this one, SF, decreased in comparison to the control. And again, no flipping stats carried out, so you can't tell whether it's significant. And the other thing that I noticed, they've only done shoot growth. why would they not do root growth as well? Because obviously the development of roots is pretty important to the productivity of plant, you know, of the plant. So anyway, so this is the mark scheme, and here is where I got the marks. Number one, actually specifying the ones that grew more. Oh, sorry, this is for saying that the control increased, and this is for saying that this one decreased. No stats got me a mark as well. For AMF, I got the mark for specifying the two that increased, and for specifying the one that decreased. And I also spotted that only shoot growth had been measured. So, you know, this was a 4-mark question. I actually, oh my goodness, ended up getting 6 marks. Oh my goodness. Anyway, only got 4, obviously. Okay, moving on. Why isn't my little pen working? Okay, let's just do this. Oh, great. Here we go. Using the E to the power X button on your calculator, determine the rate of shoot biomass production in grams per day for the control plot in soil with normal phosphate concentration. So in other words, this bar here. So log E equals 2.4. Log E to the X equals 2.4, because look, log E 2.4. So we want to know what the number X is. We want to know the actual number. So if you put 2.4 and then press E to the power X in your calculator, you will get this number. Notice I haven't bothered putting all the places in. I haven't rounded up or down, because I'm holding this number in my calculator. So this is the number of plants, oh, sorry, no, this is the number of grams that were, that the plant grew by. So we've got to work it out in rates per day. And I think, was it 20 days? Oh no, 20 weeks. It was 20 weeks, wasn't it? So now you've got to do it. You've got to divide this number by the number of days, which is 20 weeks, obviously times 7. Which equals this number of grams per day. Hopefully that makes sense, and I got the marks. Quick shout out for my website. I have got all of the videos that cover the whole A-level syllabus for both OCRA and AQA on my website. I am absolutely committed to making A-level biology accessible to every student, regardless of how much extra money there is in the household. I'm trying really hard to do it. This website is the way that I carry on doing stuff on YouTube, because YouTube pays me nothing relatively, because there's only 60,000 odd A-level biologists in the UK, and let's face it, that's really my target audience. You're a little niche group, you are, you guys. So to subscribe to all of the 2 years videos for a whole year, 50 quid. Just for the AS ones for a whole year, 25 quid. You can earn your money back if there isn't a lot of money in the household. If you can get the money together, if you recommend my website to your friends and they buy, you get 10% commission on what they buy. And there's also a freebie as well. So, you know, do at least go and have a look at it. I hope that's useful. Question 6, we're about halfway through. Each year, a few people with type 1 diabetes are given a pancreas transplant. Excuse me. Not used to treat people with type 2 diabetes. Why not? Well, type 2 diabetics are type 2 diabetics, their pancreas still produces insulin, so they don't need a pancreas transplant. The other thing is that an awful lot of type 2 diabetics, they are type 2 diabetics because the cells in their body are not responsive to the insulin. It's not because their pancreas isn't producing any. So those, oh, I've got that the wrong way around, hey ho. Here are my answers. Here's the mark scheme and the examiner's comments, and here are the 2 marks. The pancreas produces the hormone insulin. Put a tick in the box next to the statement that describes incorrectly the action of insulin. Number 1, activates enzymes to turn glucose into glycogen. Absolutely, because insulin brings blood glucose down, so it's taking glucose out of the blood by turning it into glycogen. Controls the uptake of glucose by regulating the inclusion of tranal proteins into the cell surface membranes of target cells. Absolutely does. Attaches to receptors on the surface of target cells. Yes, insulin does. And finally, and I can't read it, activates enzymes to turn glycerol into glucose. No, that is glucagon. So here's my answer. There's the answer. So I got the mark. Scientists investigated the use of induced pluripotent stem cells to treat type 1 diabetic mice. They used 4 transcription factors to reprogram skin cells into iPS cells. The scientists then stimulated the in vitro differentiation of iPS cells into pancreatic cells. Yeah, fair enough. They set up 3 experimental groups. So I have marked these groups on the graph. Group A, dash line, is they're diabetic and they have the iPS treatment. Group B is diabetic and untreated. Look at their glucose levels. And group C is no diabetes and therefore no treatment. Okay, this group, group C, is acting as the control. Yeah, you're going to compare this group with the other 2 to make sure that the active ingredient, the iPS cells, and the presence of diabetes is actually what causes the change in the numbers, in the glucose. Suggest how transcription factors can reprogram cells to form iPS cells. So here initially, I remember thinking, so this is about how do transcription factors reprogram cells to iPS cells? So my headline answer was well, the transcription factors switch on and switch off specific genes, but you're not supposed to know any more than that about what transcription factors do in terms of forming iPS cells. But you are taught about how transcription factors work. And so I kind of wondered whether I'd got my first mark by saying, oh, they switch on and off genes. But then I'm kind of going, okay, I now need more A-level detail to get this second mark. And that's why I said they bind to the promoter region, causing or inhibiting transcription. Here's the mark scheme. And in fact, it was my second sentence that got me the marks. So marked 1.2. Okay, so what is... Oh, that's annoying. Oh, that's annoying. It hasn't come up as I hoped it would. Anyway, using all the information provided, here we go again, evaluate the use of iPS cells to treat type 1 diabetes in humans. Okay, so evaluate pros and cons, looking for evidence that supports and goes against the claim in the data, and then when you can't say anything more about the data, critique the method. So the first thing I noticed, because I've looked at enough of these questions, the flip-in experiment has been done in mice, and they're now saying, evaluate this for use in humans. Well, one glaringly obvious thing to write in is, duh, the experiment's been done in mice, not humans, okay? But I'm going to use the pattern, the way that I've written it, I've used the pattern that I'm advising you. So look at the data, critique the experiment. So for this, Well, the diabetic mice treated with IPS had fasting glucose readings very similar to mice without diabetes and untreated. Untreated diabetic mice had glucose concentrations, and I did a rough calculation here because I didn't know whether they'd want numbers, 4 times that of control treated mice. There's a little hint. They sometimes do like you to just kind of go, hey, it's doubled, it's quadrupled, whatever. Okay, so I just kind of went, okay, this is about 150 and we've gone up to above 600. It's gone up 4 times. So against, this was done in mice, not humans. Results could be different in humans. And then I go on to critique the experiment, because to be honest, the data is kind of going, whoa, hey, use IPS, yeah? There isn't really anything against the argument. There isn't anything against it in the data. So now I'm critiquing. Only 30 mice per group, more needed. Only 1 investigation seems to be done. They haven't said it was repeated, so don't assume that it was. So there are my standard answers. So here are the mark points that I got. 1, 2, and 3. And did I not get 4? No, I didn't get, right, where did I? So I didn't get mark point 4. Oh, right, yes, I missed. This is a really common one that I miss. Basically that the trial doesn't go on long enough. And actually a really good thing for you guys to remember, is this enough time? Is 12 weeks really enough time to know that this is going to work in mice, let alone flipping humans? We're really hoping this is only going to work for 3 months? No, we're going to hope that it works for much longer than that. So, yeah, valid point. So question 7, what is meant by the term phenotype? The key here is to remember that phenotype are the characteristics shown as a result of both the allele combination, big A, little b, big B, little b, whatever, but also the flipping environment. I know that, of course, we always talk about phenotype, nearly always, in the context of that inheritance topic, but remember that it's the environment as well as genetic. And I as soon as I saw this, I kind of knew where the marks were going to be. So I got the marks. Okay. Inheritance of fruit color in summer squash plants are controlled by 2 genes, A and B. Each gene has 2 alleles. Okay. So we're just doing dominant recessive here. Shows the interaction of these 2 genes in controlling fruit color in summer squash plants. So the dominant allele stops white turning into green. And the dominant allele on this side causes green to turn into yellow. Okay, name the type of gene interaction. Well, this is epistasis, because if the alleles for enzyme 1 are little a, it masks the effect of enzyme 2. Doesn't matter what enzyme 2 is, because we don't get the green pigment. We're only getting white. Yeah, if we get, sorry, I've just said that the wrong way around. Sorry, let me just explain that again. So this is epistasis because if you have the dominant allele, it inhibits the white turning into green, and therefore the dominant allele masks the effect of enzyme 2. Okay, hopefully that makes sense. So if you get the dominant allele here, it really doesn't matter what the allele is for gene B, you're going to get white summer squash. Hopefully that makes sense. So epistasis, I got the mark there. What fruit color would you expect the following genotypes to have? So big A, little b, little b. Well, big A, it's not going to turn green. And if it doesn't turn green, it cannot turn yellow. Yeah, notice that this is a sequential pathway. It isn't kind of going white to green or white to yellow. You can't jump from white to yellow. You've got to go via green. So if you can't get to green, you ain't getting to yellow. Hopefully that makes sense. So little a, big A, little b would be white. Little a, big B would be yellow because you've got the enzyme 1 happening to turn it green, and then you've got the dominant allele here to make the yellow. So here we've got that, and you had to get both right to get the mark. They are not linked. Okay, fine. Complete the genetic diagram to show all the possible genotypes and the ratio of phenotypes expected in the offspring of this cross. So here is my Punnett square. They've nicely given me some space, which is very kind. Notice that the only allele, the only gamete type that could be made here is little a, little b, but there are 4 gamete types that could be made from this parent. It could be this, it could be this, it could be this, and it could be this. Hopefully that makes sense. Hence those 4 there. And therefore, these are the genotypes of the offspring, which I am now going to copy and put there. The phenotypes of the offspring, let's go back to the diagram. Big A, right, we're going to green. Dominant B, oh, sorry, big A, we're not going to green, we're only getting white. So this would be white, this one would be white, this one would be yellow, and this one would be green. Okay, so we've got those there. Ratio of the phenotypes, 2 to 1 to 1. Hopefully that makes sense, because we're doing the ratio of the phenotypes, not the genotype. So you can't write 1 to 1 to 1 to 1, because you've got to take into account that there's 2 genotypes that give rise to white phenotypes. Okay, all right, so I got the 3 marks there, up here. Okay, Hardy-Weinberg, love a little bit of Hardy-Weinberg. Population of summer squash plants produced only green and yellow fruit. The percentage of plants producing yellow fruit. So we've only got green and yellow fruit. So we're only interested in the B allele now. Okay, so therefore, does it make sense that these are the possible genotypes that are going to get us either yellow or green? We're either going to have, this dash here means that it can be either a big B or a little b. That's what this underscore means. Doesn't matter what this second allele is. Or this for green. So we're told that 36%, so as soon as you're told phenotypes, you are going to be using the longer Hardy-Weinberg equation, because p squared plus 2pq plus q squared equals 1 is referring to genotypes, therefore phenotypes. Because this is a dominant allele, we're talking about big B here, because this phenotype, the yellow phenotype, requires at least one dominant allele for the B gene, 0.36 equals these terms in that long Hardy-Weinberg equation, p squared plus 2pq. Therefore, does it make sense that q squared equals 0.64? Now, here's my tip to you. Whenever I realize that they've given me the number for the dominant phenotype, I can't be bothered to rearrange this equation in an exam. And because p squared plus 2pq plus q squared equals 1, if they've given us p squared plus 2pq, you can immediately flip it and go, right, therefore I know what q squared is. And I know that q squared is 0.64. And gloriously, 0.64 is a square number, which is rather nice. And therefore q, you'd square root it, square root 0.64, you get 0.8. Therefore, using the other Hardy-Weinberg equation, P plus Q equals 1, P equals 0.2. And then you can do your whole heterozygotes, is 2PQ, 2 times 0.2 times 0.8, which equals 0.32, but they want it as a percentage. So we're timesing it by 100 to get 32%. The crucial thing in Hardy-Weinberg is to notice that the information that they give you. If they give you phenotype information, you have got to go straight to picking out the appropriate term in the long Hardy-Weinberg equation. They nearly always give you the positive phenotype. And therefore, I just kind of go, so the positive phenotype for the B gene is yellow. And I just kind of go, oh, stuff that. I'm just going to kind of take 0.36 away from 1 to give me Q squared. And that makes the sums very much easier. You square that number, you square root that number to get Q. You then use P plus Q equals 1 to get P. And therefore, you can work out the proportion of heterozygotes by going back to using that 2PQ term in that long equation. Then the crucial thing is to notice, I said that number one, you notice the information that they give you, but then notice the form that they want the answer in. And they don't want it as a proportion or a frequency, a decimal. They want it as a percentage. So you've got to times it by 100. AI, yes. Oh, you study OCR, not AQA. Go and have a cup of tea then. The only thing that I would say is paper 1, A-level, AQA paper 1s test the year 12 knowledge, which will pretty much be the same in OCR as it will be in AQA, pretty much in most A-levels, can I be honest. So actually, AQA paper 1s are quite good tests of your year 12 knowledge to an A-level standard. And if you know your syllabus, you will probably recognise when you look at the mark scheme, the bits that you don't need to know. Paper 2 for AQA tests the year 13 knowledge. Again, useful for other syllabuses to know that. Okay, all right, so I've got the 2 marks here. Enough about Hardy-Weinberg. We're getting there. Oh, that's annoying. I've put the mark scheme up. Anyway, one way to detect and measure accurately the amount of RNA in a tissue sample is by RT-PCR, reverse transcriptase PCR reaction. Uses this just looks like mostly looks like the ingredients that you need for PCR, except for this baby here, or maybe fluorescent dye, not necessarily always. Okay, and here is the method. The amount of DNA amplified by PCR depends on the amount of RNA in the sample. Fair enough. The dye only fluoresces when bound to DNA, so you're only spotting the DNA. Yep, the intensity of the fluorescence increases as the PCR products accumulate. Explain the role of reverse transcriptase in RT-PCR. Well, reverse transcriptase, what is the role of reverse transcriptase in viruses? That is to turn RNA into DNA so that the host cell then replicates the viral genetic material. And that is the same in this thing here. So it's to turn the RNA into, now I'm being precise here, into complementary DNA. In other words, the reverse transcriptase only turns that single-stranded RNA molecule, you could have said mRNA, absolutely fine, it turns it into single-stranded DNA. You then need a primer and DNA polymerase, i.e. PCR stuff, to make the double-stranded DNA. So I was precise, but you didn't need those little letters, so got the mark. Explain the role of DNA polymerase in here. Well, here was me just kind of going, what is the role of DNA polymerase? It bonds the sugar phosphate backbone using phosphodiester bonds. And I didn't get the mark because they really wanted, for me, I had to say nucleotides because, now here's the thing, because they actually, and this was me missing the clue in the question, they asked it in PCR. Mind you, it's the same in DNA replication, you're bonding the nucleotides. But it's probably about applying this to the PCR technique because in your ingredients list are those nucleotides. I didn't get the mark. Okay, any DNA in the sample is hydrolyzed by enzymes before the sample is added to the reaction material. Well, yes, it's got to be, hasn't it? Because this, if you have any spare DNA in your sample, that DNA is going to be PCR'd immediately. What this is trying to do is really kind of turn the RNA into DNA and then PCR the DNA. So you certainly don't want any spare DNA in your sample. So that this DNA is not amplified by PCR, only the DNA produced from RNA in the sample. Hopefully that makes sense. And then I kind of went, oh, I don't know if I've really nailed this yet. And so I then said, let's really make sure they know why this DNA should be hydrolyzed, because hydrolyzed DNA cannot be replicated by PCR. So that's what I wrote. Here's the mark scheme to remove any DNA present. Now, I'm not sure that what I've written here is enough for mark point 1. I didn't give myself the mark. This was a classic case of, in my view, I'm kind of going, oh my goodness, I've nearly said, I nearly said exactly what the mark scheme required, but not quite. I didn't talk about it being removed. I didn't talk about it being destroyed. I just said it couldn't be replicated by PCR. And I'm not sure that was enough. So therefore, I only gave myself mark point 2. So we have 2 different samples here. Intensity of fluorescence, exponential increase, not surprisingly, because every cycle of PCR doubles the amount of DNA being produced. Hopefully that makes sense. A quantitative comparison can be made of the amount of RNA in samples A and B. This involves determining the number of cycles required to reach 50% maximum concentration of DNA, brackets big C. The amount of RNA in a sample can be measured as 1 over big C. Use this information to calculate the ratio for RNA content in sample A to the RNA content in sample B. Well, we've been told that RNA content is 1 over big C, and C is 50% maximum concentration of DNA, 50% of the maximum concentration. So here is the maximum concentration. This is our measure, the intensity of fluorescence. 0.48. So 50% of 0.48 is 0.24, which comes along here and gives me 19 cycles for A and 28 cycles for B. This involves determining the number of cycles required to reach 50%. That's the number of cycles required to reach 50% is your C number. So now I'm doing 1 over, I could do some water, 1 over 19 for sample A, 1 over 28 for sample B, which equals these numbers. So I could have just written this to this as the ratio, but that is not good enough at A level. You need to make this second number 1. All right. And that is the convention. You always make this second number. So in this case, B, one number for sample B, 1. And therefore you divide both numbers by this B number, because obviously this number divided by this number is 1. This number divided by this number is something bigger than 1, which is 1.47. So there is your ratio. So there's mark point, and I got the 2 marks. Notice here, ratio not expressed to 1. So if I had just written 1.47, oh, that's interesting. If I had just written 1.47, I would have only got 1 mark, which I think is slightly unfair, because that is a convention as well. Anyway, so yes. So we could have said, so now can you see that if we talked about this ratio, A to B increasing, what we're really meaning is that the ratio to B being 1 increases. Think of your surface area to volume ratio thing and how that increases. You're always making that second number 1. That's what you're talking about when you say, oh, the surface area to volume ratio has increased. Yeah, you're keeping that second number as 1. Hopefully that makes sense. And hopefully that explains why the convention is that when a ratio is written, the second number is always 1. Suggest one reason why DNA replication stops in the polymerase chain reaction. Well, you could run out of flipping nucleotides. That's, so yeah, but I would have got the mark for that. I'm pretty sure that running out of something is clear enough English to say that they're used up. Scientists have used this method to detect the presence of different RNA viruses in patients suffering from respiratory diseases. Ooh, coronavirus, can I just say? This is what happens. The scientists produced a variety of primers for this procedure. That is because the RNA of the viruses will have different base sequences because they're different flipping viruses, and therefore they will need different primers. And here was suddenly I realized where the second mark point was, because primers bond by complementary base pairing. So there we go. There's my answer. Here are here's the mark scheme. So because I talked about complementary, really easy to have only got 1 mark here and just said, base sequences differ, therefore different primers required. But hopefully when you're writing that second sentence, can you see that you might not feel like you're giving enough A-level detail for that second mark, and you would be right, and I hope that makes sense. What is a gene pool? Can I just say, it is not the numbers of different genes in a species, because species have the same genes. It is the number of different alleles in a population of a species. Yeah, think about that. Yeah, we talk about populations. If we've got isolated populations and the gene pool in one population being small, what we're saying is the number of alleles in that population. Hopefully that makes sense. So that's the mark there. Oh, sorry. Lord Howe Island in the Tasman Sea, who knew, possesses 2 species of palm tree which have arisen via sympatric speciation. So here am I thinking this is not geographical isolation. The 2 species diverge from each other 6.5 million years ago. The flowering times of the two species are different. Using this information suggests how these two species of palm trees arose by sympatric speciation. So I used my knowledge of natural selection to write, in effect, a standard answer for speciation and applying it to the fact as to why these species differ, the characteristic that we're told about that caused them to differ, i.e. flowering times are different. So here is my answer. Let me point out the classic things to write. So, because speciation happens by natural selection, yeah? Variation in the population because of mutations. Classic thing to write at the start, which the mutations form different alleles. Mutations, changes in the DNA, therefore different alleles. These different alleles cause the palm trees to flower at various times of the year. I was actually kind of going, hey, there is now a range of flowering times in this whole population. But here's the crucial things. At two different flowering times, there was clearly a selective advantage. Can I just say, this sentence here, this one is the one that was probably I didn't need to put. I'm just emphasizing that we've now got a variety of flowering times. However, the crucial thing is that two of those times in that range, there was clearly an advantage in the environment that caused the individuals that had those particular flowering times to be more likely to survive and reproduce. Yeah, and I wasn't sure whether I would need an extra bit of detail application to the question, this bit here, the presence of specific pollinators, to explain why these two particular flowering times might lead to increased survival and reproduction. I'm going to point out again, if you ever write the word survive when you're talking about natural selection, you've got to write the word reproduce, because in terms of natural selection, couldn't give a monkeys if an individual survives. If they don't reproduce, they ain't passing any alleles on. So they cause the two populations to survive more than the palm trees flowering at the other times. These advantageous alleles, i.e. the ones that caused the different flowering time, the different flowering, the ones that increase the survival and reproduction, are then passed down to future generations. When finally two individuals from these two populations could no longer breed to produce fertile offspring, you've got two species. Here was the mark scheme. 5 marks, and I only got 3 of them, I think. Maybe even 2. Did I get 3? I got 3. I got 3. So where did I go wrong? I basically described speciation. What I failed to do was to show that I knew what sympatric speciation was, which was frankly a bit stupid. I did kick myself for missing this. So but notice that these ones, this is a classic description of speciation. All right, but of course, I didn't talk about isolation. I didn't mention, which was stupid, the fact that even though they lived in the same environment, they were reproductively isolated. And I was daft that I didn't spell out that sympatric speciation means that there was no geographical isolation. They were in the same environment. And I didn't think of writing disruptive natural selection. So I ended up with 3 out of the 4 marks, which, hey ho. Finally, question 10. Here we go. We're on the home straight now. Alzheimer's, non-reversible brain disorder that develops over a number of years. Start of 2014, number of Americans had it with 5.4 million. Every 30 seconds, another person in America develops AD. Wow. In the brain of AD sufferers, there is a lower concentration of acetylcholine. This affects communication between cells. That's not very surprising given our knowledge of synapses, and initially results in memory loss and confusion. They're given donepezil, and this inhibits the enzyme acetylcholine esterase, which of course is the one that breaks down acetylcholine so that it can be recycled by the synapse, as it were. But if it inhibits that acetylcholine esterase, it's going to keep more acetylcholine there. That's what my brain was thinking. This gene mutation on chromosome 14 causes early onset AD at 49 years. I should have highlighted this, yeah. The age at which the mutation occurs varies, okay. Yaramul, isolated region of the Andes, okay. Population, highest frequency of the mutation in the world. Traced back to a common ancestor in the 17th century, natural selection has not reduced the frequency of the mutation. It's autosomally dominant. At this point, your brain is beginning to explode with the amount of information that you're being given. Involves a change in the triplet 280 from GAA to GCA. I should have also highlighted this. I missed that bit. Scientists analysed chromosome 14 from 102 individuals. They recorded a sample size of 204. Yeah. Detected 75 mutations, but only 74 cases. And I'm already kind of going, yeah, I think I can work out why. The scientists identified individuals with the mutation by whole genome sequencing. They had decided that a DNA probe would not be a suitable method. Okay. Assuming nobody with Alzheimer's died in 2014, calculate the annual percentage increase in 2014. Well, we start off with 5.4 million. Every 30 seconds, the number goes up by 1. So we've got to work out how many 30 seconds there are in a year. So 1 per 30 seconds for 365 days. So that means 365 times by 24, that gives you the number of hours, times by 60 gives you the number of minutes, times by 2, because it's every 30 seconds, gives you 1,051,200. So there's our increase. And there's the increase, and we started with 5.4 million, and we want our increase as a proportion of what we started from. So here is the calculation, and that ends up being, and I now can't read that, is it 19.5? I can't, yeah, 19.5%. So I got the 2 marks. Notice that if you, because the answer on the calculator was 19.4666, I think, therefore the correct rounding was absolutely going to be 19.47. If you didn't round correctly, you only got 1 mark, then kind of fair enough. This number here is actually, if you'd only accounted for the next 364 days, not 365. Okay, so explain how donepezil could improve communication between nerve cells. So this is what I wrote. because it inhibits acetylcholine esterase, I've now given them an abbreviation, there will be less acetylcholine broken down in AD patients and therefore more acetylcholine to bind to receptors on the sodium ion channels on the postsynaptic membrane of the synapse, causing depolarization. So less acetylcholine broken down, yep, I got that mark. Acetylcholine binds to receptors, yep, I got that mark. Sodium ions causing depolarization, got that mark. So there we go. Right, sorry. Suggest and explain why 2 reasons why there's a high frequency of mutation in Yaramil. Well, it is isolated, yeah, and therefore this population, which has got this high percentage of this mutation, doesn't reproduce with other populations, which is what, oh, no, I didn't put that. Yes, okay, so that's what I put for the first one, historically isolated, and therefore population doesn't breed with other populations. And 2, I put because it's a dominant allele, only one required for the characteristic to show. I misread the question. Here's the answer. Yeah, nothing to do with dominant allele. Notice I misread it and I was thinking that it was talking about the frequency of the disease, i.e. the characteristic being shown, not the mutation, the allele. All right, so only got 1 mark, and rightly so. I completely went down the wrong line because I misread the question. Explain why natural selection has not reduced the frequency of the mutation in the population. This was really tricky. And I did realise, because of my background knowledge of teaching about Huntington's disease, which is a dominant allele disease at GCSE in previous years, and I realised that the same thing occurs here. Huntington's, these diseases come about, they appear, the characteristic appears at an age after reproduction has already happened. The characteristic doesn't show at birth. They don't have the disease at birth, which of course might then determine whether that individual goes on to reproduce. But they have reproduced before they know they've got the disease. They've already passed on the allele. So therefore, natural selection will play no part. So because the mean age of onset of AD is 49 years and reproduction will have occurred before then. So I got those marks. This was hard. This was a hard question. Final page. The age at which the mutation is expressed to cause AD can vary. Suggest and explain 1 reason for this. I can't remember what. Oh, yes, of course. Do you know, no wonder my mind went blank. Can vary. So I had to stop and think about this. This was not a knee-jerk, oh, this is easy, this is a response. Why is it expressed at different times? The only reason I could think of was because of epigenetic changes to the DNA as a result of environmental changes that obviously change the methylation. Now, I went down the line of, okay, if the mutation, I know, is it, have I got this right? The mutation causes there to be a lower, I think causes there to be a lower concentration of acetylcholine esterase. So I said, condition causing variation in the methylation of the acetylcholine. Oh, I should have said acetylcholine esterase gene. Sorry, miswritten that. This would decrease transcription, therefore decreasing the concentration of acetylcholine esterase. Didn't need that level of detail. Just needed to know epigenetics, environmental factor. So I got that, environmental conditions, caused methylation of the gene. That's all I needed to know. And actually, I did write this wrong, but I don't think it would have lost me the mark. I think I still would have got the mark. One scientific study which analysed chromosome 14 involved 102 individuals. The scientists recorded a sample size of 204. I have clicked as to what is going on here. In this sample, they detected 75 mutations. Remember, it's dominant, but only 74 potential AD cases. Well, you've got to suggest explanation for the figures. So my answer explained why the 204 and then why the 74 cases rather than 75. Well, the 204 is because if you've got 102 individuals, each one of those had the potential to carry a maximum of 2 mutations because they're going to have 2 alleles of that gene. So because this mutation is dominant, only 1 allele is required. There are 75 mutations, but only 74 cases because one of them is homozygous. And I think homozygous, I knew I meant homozygous dominant. I did say it was dominant. So I think I would have got, one person was homozygous dominant, has 2 dominant alleles. Now, because I said it was dominant and it's homozygous in the same sentence, I think I would have got the mark points 1 and 2 there. Suggest why a DNA probe was not considered suitable. Well, if we read this, and I should have highlighted this, I didn't actually read it well at all. If the probe is only to see this change from GAA to GCA, well, how many flipping GCA sequences are there going to be in the whole genome? Loads of them. It's just too small a sequence to use a probe for. You have to sequence, you've just got to sequence the whole gene sequence. Because the mutated triplet sequence would be found in many other places on the genome, and therefore it would produce false positives. Why the DNA probe is not suitable. 2 marks, 2 points. Because it would be found in other places, and then I had to kind of go, right, what's my other mark point? Oh, because if it was used, you'd get false positives, hence, and there's the mark scheme. So I got those 2 marks. And there is the end. And I hope that has been useful. And well done, you guys who have just started watching this a little bit later, and therefore you can stop and start the video. I really hope that has been useful. And I'm not going to see you next week. I am needed to be with family next week and possibly for the next couple of weeks at least. So I am not going to be doing live streams for the next couple of weeks at all. So I think I'm going to be coming back and doing more live streams from mid-June to just build up more of a bank of them. And hopefully some of you will find them useful and helpful. From June, hey, may start focusing on AS papers rather than A2, because I would imagine that's more it's going to be year 12 students who are going to be remaining in school and working. I would imagine most year 13 students will have finished by then. I'm hoping so for their sake. Okay, see you in a few weeks' time, guys. All the very best for any assessments that you're doing. May you remember everything that you have learned. Okay, see you soon. Bye-bye.